Now that we've heard a bit more about our therapeutic development strategy, we're going to switch gears here and hear from Dr. Wendy Chung and Dr. Leah Boyle from Columbia University, who will be presenting on what we've learned from the KAND natural history study. And after this presentation, we'll have about 10 to 15 minutes for a question and answer session. So as we're watching, if you have any questions for Dr. Chung or Dr. Boyle, please put them in the Q&A box on the right of your screen so we can discuss them at the end of the session. If you're looking through the Q&A box and you see a question, you see someone's question that you really like, you can also upvote someone else's question to help move it to the top of the list if there's a specific question that we all really want answered. And so without further ado, Dr. Chung and Dr. Boyle.
Good afternoon. My name is Leah Boyle, and I'm a researcher in Wendy Chung's lab at Columbia University. Today, I'm going to talk with you about some of what we've learned so far from the KAND natural history study. Here at the Chung lab, we're dedicated to all aspects of KAND research. And I like to start with this slide because it gives a big picture overview of what we're working on here in the lab. And because it highlights the point that you, our patients and families, are at the center of everything we do. Today, I'm going to focus on disease characterization, and in particular, focus on what we've learned from the natural history study. In my talk today, after a quick trip down memory lane, we're going to start by discussing the purpose of a natural history study. Next, I'll do a brief review of what I mean when I talk about KAND, and I'll review the main symptoms we see. Then, I'll share some of our new data, including a special focus on seizures. Finally, I'll conclude with our call to action. I'll let you know how each and every one of you can help us move forward with our work towards a cure. I want to start with a little bit of history. Here at the Chung lab, we've been studying KIF1A for the last 5 years. This photo is from our last family meeting in April 2017. Pardon, our first family meeting in April 2017. And this photo is of our last family meeting, taken in August 2019, only 2 years after the previous photo. I'm sad we aren't able to be together in person this year, but I'm thrilled by how our community continues to grow. And I can't imagine how large the group is going to be next time we meet in person. As of right now, we have almost 200 individuals enrolled in our natural history study worldwide, coming from 26 different countries and more states than I can count. It's important to think about why we need a natural history study. A natural history study is one tool doctors and scientists can use to better understand what a condition looks like throughout a person's lifetime. Understanding disease progression is critical to develop care guidelines and best clinical practices, as you'll hear in tomorrow's discussion with Dr. Chung. We need to understand what symptoms occur, how common those symptoms are, when they occur, and whether anything makes them better or worse. As many of you know from talking with each other, KAND can look very different for different people. The natural history study can help us understand why we see those differences. And eventually, this will help us better predict the particular challenges you or your family member are likely to experience. Finally, a clear understanding of the natural history of a disease is a critical step in clinical trial readiness. By participating in the natural history study, you can make sure that your voice is heard, and you can make sure your family is represented, both as we develop care guidelines and as we continue our work towards a treatment. One question I get frequently is what I'm talking about when I say KAND. KAND stands for KIF1A associated neurological disorder. KAND is an umbrella term to describe all disease caused by pathogenic variants in the gene KIF1A. Depending on how many variants, the type of variants, and the particular symptoms, your doctor might use different terms. Some of the most common terms we hear are NESCAV, HSN2C, SPG30, and PHO. Scientists and doctors came up with these when they were first learning about what happens when something is wrong with the KIF1A gene. Originally, people thought of these as totally different conditions, but as we're continuing to learn, there's a lot more overlap than we thought, and these particular divisions probably won't be the most helpful way to break things down. From our work so far, we think it will be most helpful to break down KAND into specific variants or groups of variants that share molecular mechanism, since there's a good chance they might benefit from similar types of treatment. For now, though, Rather than using these imprecise terms, we prefer to use the umbrella term to talk about KAND as a whole. Although we think of KAND as primarily a neurological condition, which it is, KAND can affect the entire body. We've shared some of these data with you before, but I thought it would be useful to review the big picture before we get into some of the newer details. The most common thing we've seen is developmental delay or intellectual disability, which we see in more than 90% of our cohort. Epilepsy is seen in almost half, and a smaller number of individuals have autism, ADHD, and OCD. Although not everyone has had neuroimaging, among those who have, there are a few commonly reported findings. In 35% of people, we see evidence of shrinking of the cerebellum, what we call cerebellar atrophy. In a smaller number, we see shrinking of the cortex, a different part of the brain, and in a few people, we see abnormalities of the corpus callosum. Problems with vision and eyesight are another common complaint, and we see optic nerve atrophy in just about half of our cohort. Less commonly, cortical visual impairment, strabismus, and cataracts are seen. We know some affected individuals can have stomach problems. Most commonly, this includes gastroesophageal reflux or GERD, but we also see problems with diarrhea and constipation. A small number of individuals have irregularities in external genitalia, and a few people have kidney problems. Finally, problems with enough muscles and nerves are incredibly common, with more than 80% of affected individuals having hypotonia or hypertonia or spasticity. Before I go through our new findings, I want to take a minute to talk about something we're all familiar with, the way the same symptom can show up at different times in different people. Understanding what this means is going to be important when we go through some of the new information. To do this, we're going to imagine two hypothetical people with KAND, a 7-year-old boy and a 20-year-old woman. First, let's imagine our hypothetical 20-year-old woman. Let's say she currently has epilepsy and vision problems. Say she had her first seizure at 4 years old, and she was first diagnosed with optic nerve atrophy at 10 years old. Now, let's think about our 7-year-old boy. He also has epilepsy, but he has no vision problems. And let's say he had his first seizure at 6 years old. If these were the only people we knew about at all, we'd say 100% of people have epilepsy and 50% have optic nerve atrophy. Just knowing those statistics is helpful, but even more helpful is thinking about when the symptoms first occurred. One way to visualize this is with the graph on the top right here. The x-axis represents the age in years, and the y-axis represents percent. In blue, we can see what percent of people have epilepsy, and we can see two steps here. One at 4 years old, representing when the woman had her first seizure, and one at 6 years old, when the boy had his first seizure. This graph goes all the way out to 20 years, but in this example, the boy is only 20 years old, or only 7 years old, so we don't actually have the data from 7 to 20 for him. This is where the orange line comes in. This tells us what percentage of the total cohort, in this case, 2 people, we have data on at a particular age. So here we see a dip in the orange line starting at 7 years old, and we see only half of our cohort has data at age 8. What if we only had information on these 2 people up until they were 5 years old? I've put this gray box here to show us what the graph would look like then. It would look like only half those people had epilepsy. But if we looked at the same 2 people a year later, the blue line goes up to 100%. Now, let's look at this same type of graph, except for optic nerve atrophy. In this graph, the orange line looks exactly the same as before because we're talking about the same 2 people. The blue line here gets trickier. The woman developed optic nerve atrophy at 10 years old, and she's the only person we have information on at 10 years old. So the blue line tells us that everyone at that age has that symptom. This does not mean everyone will end up with the symptom by that age. It's just a snapshot of the information we currently have. In fact, let's imagine that same 7-year-old boy grows up to 20 years old and never gets optic nerve atrophy. This is what the graph would look like then. I wanted to take the time to go through this example because it's important to keep in mind what this type of graph can tell us and what it can't. This is a useful way to visualize trends, but it's not a crystal ball. Now for the update. Much of this work was completed by Scott Robinson and Caroline Mevin, two other members of our KAND Natural History Study, who many of you already know. As I mentioned before, natural history studies are a critical way to understand what a condition looks like over time. The best way to capture this change over time is with a longitudinal study, where you follow the same person over many years and regularly collect information so you can know what changes and when. The data I'm going to share with you today are from our first follow-up survey. In order to be eligible to complete the year 1 follow-up, a participant's initial study interview had to be at least 1 year prior. For those of you who weren't eligible for the first follow-up, we have not forgotten about you. Stay tuned for more information on what you can do at the end of my presentation. Among the 74 affected individuals we received completed follow-up surveys for, the average age at the initial interview was 9.5 years old, and the average age at follow-up was just over 11. We had slightly more males than females, but both males and females had similar age distributions. Here, we can get a bigger picture of what the age distribution looks like. We have participants ranging from just over a year old to almost 40 years old, though, as you can see, most people were around 5 or 6. Here, you can see a graph like the one I showed you before. This one looking at the development of hypertonia, or spasticity. At both the initial interview and at follow-up, we asked whether the affected individual had ever been diagnosed with hypertonia or spasticity. And if the answer was yes, we asked at what age it was first seen. The gray dotted line here is at 6.5 years old. So let's look at what's happening to the blue line at that point. This blue dotted line shows us that by the time they reached 6.5 years old, about 75% of the cohort had developed spasticity. Now, let's take a closer look at the orange line. The orange dotted line shows us that at that same age of 6.5, we had information on 65% of the cohort, or just over half. This includes anyone who's at least that age or older. So overall, this slide shows us that the majority of affected individuals did develop hypertonia, and for the most part, it showed up by about 6 years old. Now, let's take a look at hypotonia. Looking at the blue line here, again, we can see that overall, the majority of the cohort also developed hypotonia. The gray dotted line here shows us that the vast majority of the cohort developed hypotonia by 3 years old. Since at that age, we have information on more than 90% of the cohort, that tells us this information is likely to be pretty representative of the cohort on a whole. Another common symptom we hear about that can be quite bothersome is excessive clumsiness or discoordination. Here, we can see that more than half of the overall cohort have problems with coordination, but it's not nearly as common as either hypertonia or hypotonia. So if this hasn't been a problem for you or your family member by around 10 years old, it's possible it might never become a problem for you. We know that problems with vision are a common complaint for affected individuals, and they can really affect a person's quality of life. This graph focuses on optic nerve atrophy, one consequence of KAND that can result in vision decline. Unlike in the last few graphs where the blue line eventually flattened out and plateaued, here we see it continues to creep up even in the older ages. Since we have much less information on individuals older than 15, it's difficult to know exactly what to make of this. But as Dr. Chung will discuss in more detail tomorrow, even as people get older, it will be important to remain vigilant and keep getting regular vision checks so you could detect optic nerve atrophy if it were to show up. Another type of vision problem that we see in KAND is cortical visual impairment, otherwise known as CVI or cortical blindness. The blue line here shows us CVI is relatively infrequent. And this looks like something where if it hasn't been a problem by the time someone is a teenager, it's possible it might not become a problem. That said, due to concerns about optic nerve atrophy, as I already mentioned, it will still be important for affected individuals to regularly get vision checks. As I previously mentioned, a common feature in KAND is the presence of abnormal neuroimaging. We know not everyone has had neuroimaging before, And it's not uncommon for people to have imaging multiple times. So here we focused on the youngest age when any neuroimaging abnormalities were identified. We can see many people who developed abnormal neuroimaging saw it by the time the affected individual turned 6. But again, this is something that it looks like in many people continues to occur for the first time, even at older ages. Here, we can see what parts of the brain abnormal neuroimaging findings occurred in. It's worth remembering that the same person can have multiple different abnormalities, so it's not an either-or. Like with before, we continue to see abnormalities of the cerebellum, in particular cerebellar atrophy, are the most common finding on neuroimaging. Finally, let's talk about seizures. I'm going to spend a bit more time on this, in part because we know how big a question this is for many of you, and we know how important it is. Much of the work I'll be presenting now is analysis completed by Dr. Jennifer Bain, a pediatric neurologist at Columbia. Dr. Bain will be joining us tomorrow for the final panel discussion, so I encourage you to direct all questions about seizures to her. On both the initial interview and the follow-up, we asked if affected individuals had ever had a seizure, and if so, how old they were when they had their first seizure. Here we see that, like before, about half of the cohort has ever had a seizure. Although it looks like for the most part, people's first seizure occurred by the time they were 9 or 10, we do see an uptick in the later teens as well. In addition to the questions we asked about seizures on the initial interview and at follow-up, we also sent out a separate survey, in particular focusing on more details on seizures. We used a survey designed by the Rare Epilepsy Network to help doctors and scientists better characterize rare conditions that cause epilepsy, like KAND. We had an amazing 95% response rate for a total of more than 100 participants. Similar with what we saw in the follow-up medical history survey, 45% of people reported at least one seizure. Looking at this larger cohort, if we focus just on the ages of people who've ever had at least one seizure, we see 2 peaks. The first peak here is in school-age children, and the second peak here is in young adults. Even more interesting than the age of who is reporting seizures is the age the seizures first occurred, graphed here. Although for many, the first seizure occurred in the first 2 years of life, as you can see, some people don't have their first seizure until their teens or even their 20s. Here, I've pointed out the average age at first seizure, 8 years old. And here, I've pointed out the median age of first seizure, 5 years old. Now, just because someone has had a single seizure does not mean they're constantly having seizures. As Dr. Bain may go into tomorrow, our hope is that everyone with a seizure is able to find the appropriate treatment that helps reduce or ideally eliminate their seizures altogether. Of the roughly half of our cohort who report ever having a seizure, only half of that group reports having a seizure in the last 6 months. Here we can see this age distribution looks relatively similar to the earlier distribution of who is having seizures, which suggests recent seizures can be a problem for individuals at all ages. In addition to understanding who is getting seizures, it's also important to know what kinds of seizures. Many individuals who have had seizures have had more than one type of seizure. The most common type of seizure we see with KAND is atypical absence, or staring spells. These are seen in almost 70% of people who get seizures. Other common types include either complex partial or focal seizures, where there is a loss of awareness during the episodes, tonic seizures, where you see stiffening, atonic seizures, where there is a loss of tone and often dropping, myoclonic seizures, which can look like brief jerks, or generalized tonic-clonic seizures, also known as grand mal seizures. As I mentioned before, our hope is that affected individuals with any seizure history will be able to find an appropriate treatment regimen to reduce or eliminate seizures. Medications are a very important tool for this, and there are 2 main types of medications. Maintenance medications, the type of medication someone takes regularly to prevent seizures, and then rescue medications, which are used when a person is actively having a seizure. We can see that most people who've ever had a seizure take maintenance medications, while only about half have ever needed to use a rescue medication. Another big question we get is about specific medications. This is a list of maintenance medications taken by people in our cohort. As you can see, there are quite a few different medications listed, and many people take or have at least tried more than one. While I wish we had a recommendation for the perfect seizure medication for KAND, right now we don't. The choice of an appropriate medication or medications for you or your child is best discussed with your family's neurologist. To conclude, I want to first review what this new information tells us. First, this reiterates some of what we already knew. Not everyone with KAND has the same challenges. This confirmed that some symptoms, like hypertonia, are more common than others, like cortical visual impairment, and that different symptoms arise at different times. We still have a lot of outstanding questions and a lot left to learn. While we're starting to understand trends in age of onset for different symptoms, we really want to understand how the condition changes over time. I did not go into specific variants at all today, but a big question is in what ways the condition might look different for different genetic variants. We have the diagram of the gene and all of the different variants on the bottom of the slide here. Finally, we really want to get a better understanding of what treatments do and do not work for KAND. Now, what we need from you. For us to learn what we need to know, we need your help. First, we need for you to keep us updated on any changes by filling out the Year 3 follow-up survey. We're planning to send this out in just over 2 months on September 7th. You'll hear much more from us by then. This will only be for people who've been in the study for more than 6 months, but everyone else is eligible. Last year, the follow-up only included an updated medical history. But this year, we will also be asking everyone to complete an updated Vineland Adaptive Behavior Scales. This is a separate online survey we will email you about. Second, we need you to keep sending us your records. The reports are very helpful for us, in particular, reports on neuroimaging, EEGs, and OCT scans. But even more importantly, for MRIs and EEGs, we need the original images, usually sent in the form of a CD or DVD. Third, we want to make sure we can keep in touch with you to keep you up to date with all the exciting progress we're making. At the end of the follow-up study survey, we will be asking for your updated contact information so we can make sure we know how to reach you. Finally, you should keep an eye out for more opportunities to participate in the future, both things you can do from home and things that you could do in person. We'll provide more details as those opportunities arise. Here, I've listed our study team, our KIP1A study email address, the best way to get in touch with a member of our research team. We will make sure to also include this in the chat. Many people within the Chung lab work on different components of the KIP1A project, but the work I'm discussing today was completed largely by our natural history team. This includes Caroline Mebin, Scott Robinson, and Laura Ham. An additional special thanks to Dr. Jennifer Bain for her assistance with interpretation of the seizure data. Most importantly, thank you to every one of you. We could not have done it without you. Thank you.
All right. Thank you, Dr. Boyle and Dr. Chung. This time, if you could come to the stage. It looks like we've got some questions to go through. And so we'll just wait for them to come. Hi, Leah. Let's see.
Hello.
I think we're just waiting on Dr. Chung. So that's going to give me an opportunity to say congratulations to Dr. Boyle for recently successfully defending your dissertation. So thank you, because I know that was a lot of work, really, for this community.
Thanks, Kat.
All right. So while we get Dr. Chung to stage, I think we could probably start with some of these questions that I might even be able to help answer. And if you have any questions, then you can use the Q&A feature to submit questions, and we can go through those as well. And I know that a lot of you, especially our families in the community, have been sending in questions in advance in these last few weeks for Dr. Chung and Dr. Boyle to address. So I see a very popular question in the Q&A is, do we know how many people are diagnosed with KAND worldwide? And I think there might be a couple different answers to that question. So Leah, do you want to take a stab at that first?
Sure, yeah. First, also, thank you for the opportunity to speak with you all both in person now and for sharing the presentation. So the question is a very good one as to how many people are diagnosed with KAND worldwide. And I'll tie this into another question somebody added also there, and how we know this. So right now, the best guess I would say is about 350 people worldwide diagnosed that we know about. And the important thing here is that we're not necessarily hearing from individual genetic testing companies. This is something that really we're hearing in our study from people who reach out to us, and then worldwide in terms of other publications out there. So this is learning from other researchers. So the 350 number is likely much smaller than the actual number, because we know both that there are people getting diagnosed who aren't necessarily written up in a publication and who don't necessarily reach out to us. And we also know there are a lot of people who have not yet received the appropriate diagnosis.
And welcome Dr. Chung to the stage as well. Thank you both for your work and taking time to answer some of our questions. So we were just getting started with one of the most popular questions has been basically, do we know how many people out there are living with KIF1A, and how do we know?
So I'm sorry, I got lost in the back room there, but Leah, I thought I heard you answering that. I think one of the things, Kat and Leah, is that we don't really know the answer is the bottom line. On the other hand, there are efforts afoot to actually get real data and answer this question. So as an example, we will start, I think you'll see in the next year or so, some studies that will be aimed at actually, in an equitable point of view, making sure that everyone can get a diagnosis by screening newborns even potentially for this condition. And by doing that in a truly population-based way, I think we're going to find that it's actually much more common than we realized, and that there are many people who are undiagnosed. And I hope in a good way we'll be able to identify folks at an earlier time point and hopefully be able to understand the condition from the get-go, as well as to help people earlier in that life course.
And I think just from the foundation perspective, I would add to it's been really hard for us as well, because we get a lot of families who contact us after they receive a genetic report. And a lot of times that doesn't come with any kind of explanation. Maybe they just see the report. Maybe they haven't been able to have an appointment with their neurologist or genetic counselor yet. And a lot of times we find that the report says it's a variant of unknown significance. And so that's also hard too when we have newly diagnosed families coming to our community and they have that variant of unknown significance on their genetic report. So can you briefly explain basically what that means?
Sure. Maybe I'll start and Leah can chime in. So just so that people know, this is still very much a work in progress. We literally ourselves were this morning at lab meeting talking amongst ourselves about a report from a very famous geneticist. I won't name any names, but even the very best, even we ourselves don't know the real answer for some of the variants that we're seeing. So very uncertain significance I call genetic purgatory. So we see a genetic variant in the gene, but most variants in genes are not bad. They don't cause any problems. They don't cause any difference in function. They just make us the wonderfully unique people that we are. But for KIF1A, there are some that we know there are particular regions of the particular types of changes that we're highly suspicious or in fact certain that they do cause problems with the gene, but there are some that we've never seen before. It helps in terms of knowing whether that particular genetic variant was inherited from a parent or whether it started brand new. And so you'll often hear us say something like, well, what were the parents' results? Or if the parents weren't tested, can you arrange to have the parents tested? Or can we arrange for the parents to be tested? That gives us a huge amount of information, because if the parent does not have it, if neither parent has this and it's new in the child, that provides a lot of evidence that it may be, it's not certain, but that it may be causing the symptoms that we're seeing. But it's not simple, and there's still a lot that we have to learn. We've come a huge way in the last 3 to 4 years, but we still don't know everything. And so that's why we have people who are great in terms of even biophysical, mechanical properties, very basic scientists who do these experiments and look and see how these motors move and whether or not they move properly, to people like us who are able to make predictions based on some very complex computational methods and machine learning of variants.
Okay, thank you. And I want to jump to another question that I think is probably the most common question we get when we have newly diagnosed families is KIF1A or KAND always progressive? Is it always degenerative? And what do those terms mean?
So maybe I'll take this one, and again, Leah, chime in for anything. I tend to use those two words synonymously in terms of progressive or degenerative. People may have specific ideas in mind, and I'm certainly glad to clarify that for people, but I tend to use those interchangeably. The real answer is we don't know. As Leah's been talking about, and as we've definitely observed, there's a huge spectrum. And for some individuals, I think we've seen over time, just as Leah was showing, changes over time and loss of ability to do things or changes in the brain over time. So for certain individuals, it's very clear that this is progressive and that we are literally seeing loss of both function, and we even see, in terms of imaging, loss of brain cells over time. Is that everyone? I don't know the answer. I don't know that we know everyone out there who has KAND. There may be people out there who have KAND that have never been diagnosed, and they're 50, 60, 70 years old and have a different trajectory than we might have anticipated if we'd seen them only at 7 or 8 years of age. So this is very, very early days. I don't mean to, sort of I don't know, obfuscate this or try and dodge people's questions. But if you had to ask my best guess, I would say my best guess at this point is that if you look at people long enough and carefully enough, there is going to be progression in the majority of individuals. But again, that's looking at people long enough and carefully enough. And over a long enough period of time. I think what we keep thinking about is, in part, as a community, we're trying to tread water in a way, trying to keep folks where they are, trying to maintain as much as they can in terms of functionality, in terms of being able to minimize seizures. So at the time that we do have treatment, if not being able to keep people where they are or even make them better over time, trying to do that as quickly as we can so that we can minimize the progression that we see.
And I'll just add to that the idea that if we watch someone long enough and look closely enough, we'll see changes. That doesn't mean that everyone will have the same changes. So there might be one person who has a progression in their vision problems. Somebody else has a progression in problems with walking. The idea that something might progress doesn't mean that everything will progress necessarily.
Lisa, I see Lisa submitted a question that actually was also submitted in advance. This question about, is it possible that CVI is something that could be underreported? And someone else had asked also about seizures, because some of these things don't seem to be black and white. Especially with the absence seizures, I feel like sometimes anecdotally we hear in the families that even our own neurologists aren't sure, because seizures aren't always black and white. And the same thing with CVI, especially it might be even hard to diagnose. So do you think those two particular symptoms, and even any others that you can think of, might be underreported in the current natural history study data?
So I'll start this, and Leah, you can chime in. So CVI, I think everyone knows, but in case someone doesn't, is cortical visual impairment. And so it's individuals who have problems with vision. It's not a problem of the eyes and wearing glasses or something like that. It's really receiving the signal in the brain. It's hard, and I think you're right, Kat, in terms of underreporting. For many of the folks in our community, you know, I think it's like, for me, like, how would I know any differently? If I can't see, how do I know that it's coming from my eyes or coming from my brain? Like, how do you know, right? I mean, you're having trouble seeing, or you're tripping or falling or running into something. Like, where's the problem? Who knows? So I'm sure you're right in terms of that being underreported, not just by people with KAND or their parents, but we as doctors too. I mean, like, like I said, how are we supposed to know where it's coming from? So I think you're right about that. For the epilepsy portion of things, especially for something as subtle as an absence seizure, again, I'm sure that's underreported. I doubt it's overreported. For other conditions, I'll have people telling me, oh, their child's just daydreaming. And, for other something else, that might be true. But in our community, what looks like daydreaming, I think, is probably more likely, not definitively, but more likely to be an absence seizure. Just to give you guys a sense, and Leah was sort of alluding to this, we've got big things planned for you guys. So I'll warn you about this in advance. We want to have, and Kat is in particular, I'm sure, going to be a great, Kat and Dom are great facilitators of this. But we need to hear from you in terms of what your priorities are, because we're getting ready to do some very big things in terms of creating a study protocol. It's going to be sort of natural history 2.0 is the way I think about it, but a more intensive natural history study for some of you that choose to do. Of course, you're not forced to do anything. And within this, part of this will likely be not just surveys that you're doing, not just talking to us on the phone, but doing some things in person, doing some things remotely, but with some devices that we might give you, really doubling down, because as we've talked about before, we need to be able to understand, when we think about a clinical trial, seeing that change, seeing that movement, seeing that we've had an effect or not. And so we're trying to think about multiple dimensions in which we can do this and measure things very quantitatively, so that we've got real hard data to be able to show early it's either having an effect in a positive way, having an effect in a negative way, or not having an effect. But we need a way of being able to say fast fail is like the way I'd be able to think about fast fail or fast success, but do it quickly so we can go on and iterate and move on to something else if we don't hit it right off the first time. So one of these things, to give you a sense, that I've thought about is actually doing EEGs from home. So it wouldn't be the same type of thing for those of you who have thought about, some of you know, I know you've done it, come into the hospital, done an overnight video EEG. We use that hideous glue to paste those electrodes and mess up your hairdo. So it's nothing like that. I just want to reassure you. But it would be something that would be much easier, where you could just put on a cap, put on a hat or something at home, but be able to have some transmission. It might not be same sort of rigor. It might not be quite as high a quality as we would get by having you come into the hospital, but on the other hand, we could do it more often, and we might be able to do it in a more naturalistic setting. In other words, the way things really are for you, rather than this artificial system we have in the hospital where you're not sleeping normally, you're not, like you're off your game and all of those things. So I won't promise today for any of the things that we're actually going to do, because it's actually predicated on what you tell us is important to you. But based on that feedback, then we will design the protocol, like I said, to meet what your needs are, what the gaps are in terms of greatest gaps, greatest things that we need to be able to address, as well as with input in terms of what's going to be a rigorous outcome measure that we can have in those trials. But that's of some things to come.
And I know we will all be very happy to contribute and make sure we're working with a good plan that works for everyone. And I think we're all really eager to get more involved in the research, and hopefully some of that at-home stuff will allow, more equitable participation in the research as well. I'm looking at the Q&A, and I see Avantika has a really great question. Considering that the KIF1A gene is a motor protein, why does it affect a patient's cognition?
I can take this. So when we talk about it as a motor protein, the way I think about it, that has to do with what the protein's doing, not what the protein does to you or in you. So the idea is cognition is really a bunch of different signals in your brain being sent from one place to another. And the KIF1A molecule is what helps move those signals. So the problem, when you have a problem with the KIF1A motor, it means that the signal isn't able to go from place A to place B. And that's a big part of cognition, is being able to have the thoughts transfer. And this is also, it's the same thing that's actually related to why there are problems with things like walking, because it's different signals that you're sending. If it's the signal from one part of the brain to another, or the signal from your brain to your big toe, all of those, it's how you get those signals from one place to another. That's what we mean when we talk about motor proteins.
Is there an answer to this question? You know, a lot of, and this actually relates to a question I think Jane submitted about this mix of hypotonia and hypertonia. I guess it's always confused me why you know, spasticity is such a problem in the lower limbs, but not necessarily, there's not the same spasticity, at least not in all the patients with, you know, arms. So does that have to relate with, you know, the motor protein and how it works, you know, carrying signals to from the brain to the parts of the body?
Yeah, so I'll start taking this. So part of, I think you actually started to answer your own question there. Part of why the legs are different from the arms is because the signal from your brain to your big toe, it's a lot longer to travel than from your brain to the end of your thumb. And one of the things that we're starting to learn about KAND is that it does seem like if you have a longer distance that a signal has to travel, that's where we see more of the problems. So I think that's some component of it. As far as the hypertonia and hypotonia, when we use those terms, we're talking about at all within the body, but exactly as you said, the spasticity in the lower limbs, you might have spasticity in one place and have hypotonia in a different place. So you can definitely have them in different places at the same time.
And I'll just note too that we do have some sessions put aside for symptoms Q&A, and we'll also have Dr. Bain there as well. So I'll hold off on any of the seizure related questions right now, and we'll dive into those tomorrow. Let's see. I know I asked earlier, whenever I saw your slide about, you know, with the graph and clumsiness, is that ataxia, or what's the medical term for that, I guess?
I'll answer as far as what the slide says, and then Dr. Chung can add for the terminology. So the reason I used clumsiness on the slide is because clumsiness is what we asked you all about. That in some cases, the answer was, yes, she's clumsy, she has ataxia. In other cases, that information wasn't necessarily provided. So in sharing the information back, we're trying to not be too specific, because ataxia does have a very particular meaning. And since that's not the question we asked, we didn't want to give that answer. But Dr. Chung can talk about what the meaning.
Yep. In the children, the technical term that I think is probably more appropriate is apraxia. And so it sounds like ataxia, but it's apraxia, PR. And what it is a problem in terms of motor planning and motor coordination. And it tends to be something that isn't KIF1A specific, but we actually see it with a lot of children who have neurogenetic conditions. And it's a problem, like I said, with coordination and planning for complex motor movements. The more you practice, the more you do it, the easier it gets, but it's still hard.
Right.
And some people may hear about that there's apraxia of speech, so you can have the same problem, sort of clumsiness of your tongue and your lips, and so making those different sounds when you're speaking can be an issue for some kiddos too.
Okay. I want to shift gears just a little bit and address some questions that were submitted in advance. But again, these are some of our most common questions as well. And it really centers around like inheritance. And so we get this question a lot. If a parent will ask, if I have a child with a KIF1A mutation that is de novo, how likely or unlikely is it that any future children would have a KIF1A mutation as well?
Yeah. I'll give the simple answer, which we know empirically to be about 1%. So even though it's something that we don't see in parents when we do the either blood sample or a cheek swab sample, we do know that about 1% of the time it can be in an egg cell or a sperm cell that gets passed down to another child. So the way I read that is it's not very frequent, but it's not zero.
And then another question about terminology. And Dr. Boyle, thank you for going over that umbrella concept in your presentation, because I know all these different terms, especially when we're comparing genetic reports, they may have different terms on it, but it all falls under that KAND umbrella. So that's helpful. We're starting to see more and more families join our community with deletions in KIF1A. And sometimes it's not just the KIF1A gene that's been deleted, but that there's other genes on that chromosome that have been deleted. So at this point, would you consider people with KIF1A deletions, if it's contributing to their symptoms, would we say that they have KAND as well?
This is something where I think it's hard to give a general answer, because a lot of this depends on things like how big the deletion is, for instance. So if it's a whole bunch of genes, then we might say this is part of it. But it's difficult if there are multiple different genes that are involved, KIF1A might not be the only answer, or KAND might not be the only answer. If you're talking about a smaller deletion just within the gene, if we know for sure that deletion is disease causing, and this goes back to what Dr. Chung was saying about how we're still learning for a lot of these variants of unknown significance. But if there's a deletion that we know is disease causing, then yes, we would consider that, we would call that KAND.
I see Cassandra has a question in response to your call to action to send you all of our records and stuff. So to answer that question, I'll let you answer that now, but I'll say we could also send out an email to the community to give them all the details about how to get their records over to you.
I'll say I was going to say, as the short answer, I also just put an email address in the chat. So that's Dr. Chung can talk about what information to send there.
Scott Robinson, as Leah alluded to, is the genetic counselor who's coordinating everything, and Scott will follow up with individuals. He'll answer the study email address, which Leah put in the chat, and be able to work with you in terms of transferring discs and other records. And we'll pay for postage or anything that's required to get us the data.
Okay, thank you. What about, let's see, from Avantika, would you consider CVI to be the same as visual processing? Do you think it relates more to vision or brain processing? And Dr. Chung, you may have answered this about CVI is really more related to the brain, right?
Right. So CVI is cortical visual impairment. Again, it literally is the back of the brain here that's responsible for receiving the signals in the eyes. There's a little bit more when it comes to visual processing that's even more than just receiving the signal. So it's even a little bit more complicated than just receiving the signal. But you can think about it as the problem is with the brain, back, sort of the brain rather than the eyes, in terms of the problem being directly with the optic nerve, for instance.
Okay. So the next question coming to the top. In a child with one unaffected copy and another affected copy, do we know if there is a difference or variation of expression determined by each of the two copies? And might this impact the outcome? And that also kind of relates to a question I was thinking about teeing up for you guys in terms of talking about this concept of, and maybe it relates, I don't know, as like KIF1A functioning as a dimer, and then also the fact that most of the KIF1A mutations that we see in our community are heterozygous, and I guess kind of the implications around that.
So a simplistic way of thinking about this is, of the two copies, they're both produced in approximately equal amounts. There are slight differences. What's implied by this, although maybe not intentionally so, is there might, big might, but people could think about either trying to upregulate or downregulate specific copies of the gene because they are on two different copies of the chromosome. We're not at the point of realizing how to do that yet, but for some other conditions, people have thought about strategies like that. And maybe there's a way of being able to use that to our advantage.
you. Let's see, we have a question. Are there resources for families who can't afford genetic testing? Is there someone who will pay for it? I will speak for the foundation first and say if anyone in the community needs a resource around getting the answers you need from genetic testing, please contact us, and we will find a way. We will find someone to work with to get you the answers that you need. But I guess Dr. Chung, from a more clinical setting, what would your answer be to that question?
Yep. So it's a little bit complicated depending on what country you're in. What I can say is that different countries have different payers. Even within the United States, we have different payers. But I think Kat is correct that one way or another, we're committed to helping you to get the answers that you need, whether it's KAND or whether it's something else. But we'll take it one off, basically. But we can help, help navigate you to those resources.
We have a few questions about microcephaly and KAND. So is there a correlation? And yeah, I guess based on the natural history data and what you've seen, what do we know about any kind of connection between KIF1A and microcephaly?
So I'll say it's not always associated. Microcephaly simply means small cephalic head, so small head. It's not always associated with microcephaly at birth. We do know that over time there can be shrinkage, and so it's not as if the head size shrinks, but within the skull itself, the volume of the brain can shrink over time. But technically, most people are measuring sort of the head circumference or your hat size or your skull size, and so when we see evidence of microcephaly, that means that that was present from very early on, as opposed to this being that progressive or degenerative portion that I was talking about before. We can see it in some individuals, and it's been very clearly documented for specific genetic changes, but it's not consistent. And again, it's kind of the story of KAND. The majority of individuals do not have microcephaly in terms of measuring their hat size or measuring their head circumference, but it can be.
Okay. I see a very important question from Rick and Emma around, unfortunately, we do have, losses in the community. And we know that, some families, they're able to make the decision to make a donation of the brain in order to advance KIF1A research for other kids. So can you talk a little bit about how that really has helped KIF1A research move it forward?
So let me just say, the gift that our families have given to the community has been, I just know, so incredibly hard, but so incredibly generous and so incredibly important for all of the kiddos and all of the folks out there with KAND. I think about them and I look at the pictures of the children and think about the angels watching over us in terms of what they continue to teach us and help us with. I'll just give you one example, is that we knew that with KAND that things were changing in the brain, and we had a sense that things were changing in the spinal cord or the brain coming out of the brain here, but going down the back and perhaps even other parts. But we didn't know that for sure. And when we were trying to think about where, if we had to give a medicine, for instance, where were we going to have to give the medicine? Just a simple question like that. Generous donation of the families, we've been able to start understanding what parts of the body are affected, what parts of, we technically call it the nervous system, are affected, and we have some sense now of which parts are most sensitive. And in terms of just literally being able to think about where we want to give a drug or where we want to give a therapy, do we need to give it here? Do we need to give it here? Do we need to give it in the big toe? It's a very, very practical question that I would not have been able to know for sure without being able to see those things. One of the challenges that I don't, although I think we've learned an enormous amount from the 3 autopsies that we've been able to perform, each time we've learned more and more. And I think to myself, I wish I had done X, Y, or Z because I didn't even think at the time to do it. It was just, it's a learning process, and I didn't even think to ask certain questions because I hadn't realized the extent of things. And so I think we continue to want to do that, and I'll just give you a certain example. And I know it's so painful and gruesome to think about, so apologies. And if you want to turn off your walk away and take a bathroom break, certainly go ahead. When we think about, again, where we want to give a treatment, I'm not sure if this is something where we need to give systemically. So we need to literally get to the entire part of the body, or do we just need to get it to the brain or the spinal cord? And in certain cases, like I said, we didn't look at the muscles very carefully. We thought this was just going to be a brain issue. We just need to get it to the brain. Now I'm starting to wonder, do we need to actually even get it to the muscles over time? Do we need to get it into the eye? Like, do we need to go this way or do we need to go this way in terms of doing it? And so there are still some questions that I don't think we know the answer to. We know, probably a good amount of what we need to do to be able to take a treatment forward. But there's still some unanswered questions because we've realized that it's more extensive than I think we first appreciated. So gosh, I hope we never ever have to do this again. I mean, if I could blink and say that we would never lose another one of our angels, then I would do that and we'd never have to have this conversation. On the other hand, I will tell you that I really feel their presence with us all the time in terms of literally touching and guiding the work that we're doing to be able to make sure that they're other friends. That's the way I think of them, as they're other friends to be able to take care of them. So for those of you who are parents or family members of those angels, I just can't tell you how much your contribution has meant to the entire community and will for generations until we do cure this.
Absolutely. And, I know we have several more questions to get through, but again, we'll have more time tomorrow to go through those as well. Right now, and maybe someone can ping me if I'm wrong, but I think we're at the top of the hour now. And so if your question wasn't addressed, we'll likely get to it tomorrow. So thank you again, Dr. Boyle and Dr. Chung for, it's really just not this conversation and your presentation, but, I think it was Sally who said it once, but dedicating your life to making our lives better. And so we really appreciate everything you do. With that, I'll kick it over to Dominique.